Hello, everyone, and welcome to the Global IMMUNOTALK. My name is Matteo Iannacone, and it is my great pleasure to introduce Antonio Lanzavecchia as today's Global Immuno speaker. Antonio really would need no introduction, as he is a true giant in immunology. I'm going, however, to say a few words, especially for the young students in the audience. Antonio was born in Italy, and he obtained a medical degree from the University of Pavia, where he also specialized in pediatrics and in infectious diseases. And from 1983 to 1999, he worked at the Basel Institute for Immunology, and since 2000 as the founding director of the Institute for Research in Biomedicine in Bellinzona, Switzerland. And very recently, he has relocated to Milan at the National Institute for Molecular Genetics, where he is continuing his productive academic career. Antonio's discoveries have really redefined many chapters of current immunology textbooks. And what is truly remarkable for me is the breadth of his scientific interests. And just to mention a few areas where he made a lasting impact are his work on antigen presentation by B cells and by dendritic cells, his studies on T cell activation and on the cellular basis of immunological memory, and the development of novel methods to isolate human monoclonal antibodies. And it's this last remarkable technological development that spurred, I think, the current focus of his laboratory, where he investigates the mechanism of antibody-mediated resistance to infectious diseases. He cleverly used high-throughput cellular screens to interrogate human memory B cells and plasma cells and to isolate potent and broadly neutralizing antibodies against a variety of targets ranging from common pathogens to emerging viruses. And these antibodies are developed both for prophylaxis as well as for treatment of infectious diseases, but also they are used as tools to produce optimal vaccine components in a process that he refers to as antibody-guided vaccine design. And besides these translational studies, the laboratory also addresses fundamental aspects of the antibody response, such as mechanisms that lead to the production of broadly neutralizing antibodies, the relationship between infection and autoimmunity, and very recently, a fascinating new mechanism for antibody diversification through templated DNA insertions that he recently discovered in the context of the antibody response to malaria. Now, in recognition for his work, Antonio received numerous prizes, including the EMBO Gold Medal, the Cloetta Prize, the Robert Koch Prize, the Sanofi Institute Pasteur Prize, and the Louis Jantet Prize. He's a member of the EMBO and a foreign associate of the US National Academy of Sciences. And finally, in addition to his academic work, is also the scientific founder of Humab's Biomed, which is now a subsidiary of Vir Biotechnology, where he's currently senior research fellow. So thanks so much, Antonio, for having accepted our invitation. We are very much looking forward to your talk. But before we start, as it is a tradition of the Global Immuno talks to ask speakers a question to get to know them a little better and perhaps to inspire the next generation of scientists. So the question we have for you is, what is the advice you would give to your 25, 30 years old self?
Well, thank you for the invitation, first of all. That's a difficult question. I can tell you what advice I found very interesting in my career. And the advice comes from, there are two pieces of advice. The one comes from Franco Celada, who was the mentor when I was a young MD. And he told me, Antonio, do not follow the fashions. Stay in the center of the field. Go for the important questions in immunology. And at the time, I was lucky, there were many questions open. For instance, TB collaboration, then dendritic cell, antigen presentation, activation, differentiation, and memory, and so on. And the second, of course, it's important to find your experimental system. And my decision was to go to study human cells, because this was easy. I was in a small lab in Italy, and you know, I could easily draw blood from my colleagues. The second piece of advice is much more specific and comes from Fritz Melchers, who was the director of the Basel Institute at the time, and was very simple. He says, have fun. And I think if you follow the real science and if you have your method to do it, you are going to have a lot of fun by, sticking to immunology and sticking to the basics.
Fantastic. Thanks so much for these inspiring words. And without any further ado, I think you can share your presentation.
Okay. Can you see my presentation?
Perfect.
Okay. All right. So I will talk about some recent work in the lab dealing with the isolation of antibodies from memory cells, and I choose to divide my talk in three parts. We can make useful antibodies. We can understand basic aspects of memory repertoire and dynamics, but as always, and this is a part of fun of our work, we can find also something totally surprising. So the story starts many years ago. At the beginning of the last century, we used horse antisera to protect against diphtheria and tetanus, and this saved thousands of lives. And this serotherapy was brought into the 60s and the 90s by developing specific human immunoglobulin. But it is only in 1998 that the first monoclonal antibodies, this was actually a mouse monoclonal antibodies, was introduced for prophylaxis of neonatal RSV infection. So the reason for this very slow development is that mice are not an optimal platform to develop monoclonal antibodies against human pathogens. And but things are changing now rapidly. In the last 20 years, several laboratories, including our own, have started to isolate monoclonal antibodies from human memory cells. And as you see here, there are now a number of antibodies that have been approved for therapy of a variety of infectious diseases, including Ebola and COVID-19. And these two antibodies have been isolated in my laboratory or by my colleague Davide Corti at UMAB's Vir Biotechnology. Now, the method that we developed in early 2000 is very simple. I mean, the idea is that there are individuals that give a particularly good antibody response to a given antigen. Now, we isolate the blood of these individuals and we screen the antibodies produced by immortalized memory B cells or by plasma cells, and we can screen these antibodies using functional assays, for instance, neutralization, and even multiple assays. So we don't need to know in advance what is the target. And this is a great advantage because we can be target agnostic. So we can essentially discover good targets without knowing a priori which are the target molecules. And then the idea is, of course, very simple. Once you have the clone, you sequence the antibodies and you produce them in a recombinant form. You can modify the Fc and then give the antibody in this novel serotherapy approach to transfer protective immunity. Now, this platform was very useful to isolate antibodies very quickly against emergent viruses. In fact, our first work dealt actually with isolating antibodies from SARS convalescent patients in 2003. This is the paper where we first described the method, and then we went on and isolated antibodies to MERS and to Ebola, and luckily through collaboration with our colleagues at the NIH, this antibody was further developed and is now used to treat the patients with Ebola, where when given early on to already infected individuals, the mortality can be reduced by more than 90%. This is we thought we were amazed by these results because it shows that antibodies can be used not only prophylactically, but also therapeutically. Now, Matteo alluded to this idea that not only the antibodies can be used, if you like, as drugs, but also as tools for vaccine design. So by mapping the specific site of where the antibody binds, you can identify the protein and you can produce it in a stabilized form so that it will induce the same type of antibodies. And I want just to make a couple of examples. So first of all, you can screen and try to identify antibodies according to for their potency. For instance, we found that the most potent antibodies that neutralize human cytomegalovirus do not bind to the fusion proteins, as one would have expected, but rather bind to this complex pentameric protein. And so we tested a pentameric vaccine in mice and found that it induces in mice neutralizing titers, which are, as you see here, almost astronomical, and at least 300-fold higher than the level of neutralizing titers developed in human sera 2 years after the infection. So if you use the antibody to identify the right target, then you can produce a more effective vaccine. And these are two other examples. These are antibodies that neutralize all influenza A viruses by binding to a conserved site in the stem of influenza hemagglutinin, or this other antibody that neutralizes 4 different viruses by binding again to a very conserved site in the F protein of these paramyxoviruses. Now, what we learned from these studies is that these antibodies can trigger very potent effector function that contributes to protection in vivo from the infection. And I should highlight that antibodies to the globular head, for instance, do not induce, do not mediate effector function. So the capacity of an antibody to trigger effector function depends very critically, not only from the isotype of the antibody, but also from the site where the antibody binds. And so these antibodies that binds on, say, on the site of the molecule are particularly good in triggering effector function. The second aspect is that because these antibodies bind to conserved sites, they're less prone to select virus escape mutants. And this, of course, is another advantage of this type of approach. Now, I want to tell you the surprising story, but really the reason to start to study the antibody response to P. falciparum, to malaria in malaria patients, is really due to the fact that we thought that this is an extremely interesting system because it's a clash between what we call two evolutionary titans, the pathogen that can use hundreds of different genes to play with the immune system, and the antibodies that can recognize different antigens on the pathogen. So, and there are now, I want to look at this P. falciparum in two ways. First of all, to look at the infection. And the infection is carried out by just very few sporozoites, which are introduced by the mosquito. And they are so few that they don't induce an effective antibody response. In fact, the sporozoid surface protein is not polymorphic, it's just monomorphic. So the idea of having a vaccine that will block the sporozoid is a very attractive idea because could confer sterilizing immunity. However, the current vaccine, which is called RTSS, has a very low efficacy in the field, around 30%. So there is ample room for improving the CSP-based vaccine. And so what we did, these are two papers, one from our group and one from the NIH group, with similar results, essentially consistent results, is that we studied the immune response of volunteers that were immunized with large amounts of sporozoites, and so mounted a response against the parasite. And we found isolated potent blocking antibodies, and we found that these antibodies have a particular properties that they bind to multiple sites on the CSP protein. There are this called NA and P repeats, but also bind to an N-terminal peptide, which is not present in the current vaccine. So the take-home message here is that by including this N-terminal peptide, we could induce more effective blocking antibodies. And in fact, the NIH group has now followed up on this initial finding and demonstrated that these antibodies isolated from these immunized volunteers can actually very efficiently protect from infection. So there is hope that we can now use antibodies for prophylaxis, but especially we can improve the vaccine by using the whole CSP rather than just this bit to induce these potent neutralizing antibodies. Now let's get instead to the blood stage parasites, because after the liver stage, the blood stage parasites are present in huge amounts. Now, at this point, the immune system recognizes the parasite, but the parasites have a way to evade the immune response because they express the so-called variant surface antigens that are thought to mediate adhesion of infected erythrocytes to endothelia. I will show you that they have another function as well. Now, it's known that antibodies to these variant surface antigens can protect from the disease. Of course, they don't protect from the infection. But, however, it's important to notice that the parasites has 200 of these VSA genes. These genes are polymorphic and are clonally expressed. So for the parasites, it's very easy to select the different variants that escape the antibody response. So our question was, can we find antibodies that broadly recognize infected erythrocytes? And so we started by taking our agnostic approach. We screened Kenyan plasma from about 500 individuals and found that some rare individuals had antibodies that could cross-agglutinate different parasites. You see here, these numbers refer to parasites isolated in the region, and this donor in particular had antibodies that could cross-agglutinate all these different parasites. So we immortalized the cells and isolated, as you see here, a bunch of antibodies, and we're very pleased to find that several of these, in fact, most of these antibodies recognize all different parasite isolates, as you see here. It's interesting to notice that these 3 antibodies are actually very specific for a particular isolate, which again tells you how polymorphic is this is the parasite, and how interesting is the finding of broadly reactive antibodies. Now, the surprise was that when we sequenced these broadly reactive antibodies, we found that all of them carry a very unusual heavy chain that carries a mutated layer 1 exon inserted between the V and the DJ. Now, layer 1 is a collagen binding inhibitory receptor, which is encoded on another chromosome, on chromosome 19. So this is the bit more in the detail of these antibodies, the V, the layer 1 exon with little intronic sequences, and then the DJ. And in the protein, of course, the layer 1 domain is appended, if you like, on the tip of the CDR3. Now, I summarize here quite a few experiments. So we found by proteomic analysis that layer 1 antibodies bind certain refins, and refins are a type of variant surface antigens. The antibodies, by binding to the refin, will opsonize the infected erythrocytes and facilitate removal of these erythrocytes. The layer 1 domain alone is sufficient for binding to the refin. You don't need the rest of the antibody. In each individual, we isolated many different cells producing antibodies, but all of them were deriving from a single clone that underwent a very high level of somatic mutations. So here there are 48 amino acid substitutions, and there are 2 types of mutations, those that abolish collagen binding and those that increase binding to the refin. And it's interesting to consider this first type of mutations because the layer 1 is, as a matter of fact, is self-reactive, it binds collagen. And so the B cells are under pressure to mutate away from this self-reactivity. This term has been named clonal redemption of the clones by Chris Goodenough. Now, the next question was how common are these layer 1 containing antibodies? So we screened the 2 additional cohorts in Tanzania and Mali, and we find that approximately 10% of the individuals have antibodies that contain layer 1, and from these individuals we isolated monoclonals. And it's interesting that in individuals that make layer 1 antibodies, this single clone that produces layer 1 containing antibodies dominates over the whole response to infected erythrocytes. So this is a remarkable example where a single cell can take over a very broad and potent antibody response. So because we have the cells that make the antibodies, we can look again at cDNA and genomic DNA. We find different types of insertions, all comprising the layer 1 exons, and in all cases, the exon appended on the CDR3. But surprisingly, again, we find other cases where the insertion was actually taking place in the switch region. And here, what is inserted in the exon with intronic sequences so that the exon could be spliced in, and the domain, layer 1 domain, now will be positioned between the VH and the CH1. And I will show you later the structure. So, at this point, we were really intrigued. So, are there other examples of templated inserts of antibodies? Is this a new mechanism by which you can generate antibody diversity? Is this, or is it limited to layer 1 for whatever reason? So to address this question, we took 2 approaches. First, we took an unbiased approach. So we started to sequence the switch region of antibodies in European blood donors, because we wanted to look whether this phenomenon could take place even in the absence of malaria infection. And we find that, yes, these immunoglobulin genes contain fragments, templated inserts that derive primarily from genic regions and very often contain whole exons. And you see here, these inserts come essentially from just whole chromosomes. And if you're interested to understand more about this mechanism, I should point out a paper that just was published by Catherine De La Rosa, who was the first author in our Nature 2017 paper. Now she has now described in very great details the nature and the mechanism of these, potentially the mechanism of these insertions. So then what about other type of antibodies? And then we made what we can call an educated guess. So what about other inhibitory receptors? And so we considered the LILRB1, which is a class 1 binding inhibitory receptors, and search again this Malian cohort for the presence of antibodies that contain LILRB1. We found individuals with such antibodies, and in these 3 individuals, we find insertions in the switch region of LILRB1 the genomic insert that encode domain 3 and 4 or domain 3. And by proteomic analysis, once again, we identified that these LILRB1 inserts recognize different regions. And now I come to what I consider from the biological point of view a very exciting finding. At least it was super exciting for us. Just think of the problem of the parasites. So first of all, the parasites targets inhibitory receptor, and this has been a new concept because it was thought that the parasite would just stick to the endothelia, but now we find that it wants to trigger inhibitory receptors. The escape from antibodies is facilitated by the fact that these refins are polymorphic and they're also clonally expressed. But the layer 1 and LILRB1 containing antibodies can recognize multiple genes and multiple refins and multiple alleles. In fact, you see here that many different refins bind to either layer 1 or LILRB1. And we are now committed to try to complete this analysis to really map the interaction between the parasites and inhibitory receptors. Now, a couple of slides to finish off on the amazing structure that was resolved in the laboratory of Peter Kwong. So these are two different refins that bind to layer 1 or LILRB1. We call it the razor cartridge and handle razor model of binding. And this is the structure of the inserts in the switch region. You see here that in this FAB fragment that contain LILRB1 insertions has very unusual structure. The VH and VL and CH and CH1 and CL domain maintain the original structure, and what happens is that the molecules open up around this hinge to accommodate the insertion of the two LILRB1 D3 and D4 domains that bind to the refin. Now, with this, I think we have learned something from some immunology from studying the immune response to pifilcyprum. We have some hints on how to improve the antibody. We know something about the pathophysiology, the fact that the pathogens trigger inhibitory receptor, and this, of course, is for a good reason. And we have shed some light on the new mechanism of antibody diversification, and we call this receptor-based antibodies. Now, in the last 15 minutes or so, I want to now switch to a more basic aspect of memory cells, and this is, I think, is still, I would say, ongoing work, although the first part has been recently published. This is work has been done primarily by a very talented postdoc, Ganesh Phad. And our question was to systematically analyze memory B cells and circulating plasmablasts using a single-cell RNA-seq. And so the basic question was, what's the structure of the memory repertoire, the size of the clones, the isotypes, the isotype usage, how stable are memory cells, but more importantly, what's the dynamics of memory cells, and what is the specificity of circulating plasmablasts, which are continuously produced by the immune system. And so the experimental approach, very simple, serial samples from 2 individuals that we followed over many years. We have paired the VH, VL, and isotype sequences we use in the 10x platform. We reconstruct clonal families. From the same individuals, we isolate antigen-specific antibodies so we can know the specificity of some of these families. And we have put all the data accessible through an interactive web-based platform, the RepSeq playground, that I hope some of you may be interested to play with. So the first finding, I mean, we call this mandala plots, is that if you look at the structure of the memory B cell pool in these 2 individuals, you see that there are at the center, there are some very large clonal families. But you see that in both individuals, these clonal families are green, so they are mostly IgM. And this was a surprise because I would have expected, that the most expanded clones would be probably IgG. We are vaccinated, we see viruses all the time. Instead, the most expanded clones are definitely IgM. And you see here examples of these clones in this waterfall plot. You see the largest clones here have a clonal size in the blood in the order, we estimate, of 5 times 10 to the 8 cells from a single clone. And you see here an example of these clones. You see how diversified they are. And these plots, I should just remind you that if you see vertical cells aligned vertically, is because they have exactly the same sequence. Nonetheless, you see that here there is quite some diversification before clonal expansion. So these expanded families, there are also expanded families of IgG2 and IgA, but once again, the surprise is that these are isotypes which are relatively considered to be T-independent. So the largest clonotypes in the memory pool appear to be T-independent rather than T-dependent. So how can we learn about the specificity of these cells? So what we are doing now, and this is a follow-up from the paper, we isolate systematically IgA antibodies to pathogens and commensals and search for the corresponding family in the playground database. And this is an example here where we have isolated an antibodies to Staph aureus, and there is a perfect match with this particular clone and in this very large family. And you see here the binding. But the surprising finding is that when we now express these two other antibodies from the same clonal family, these antibodies do not bind to Staph aureus. So I think these findings, and that of course one had to be extended to many other examples, but suggest that diversification through somatic mutation occurs before selection by antigen. I think this is a strong statement, and I would I will caution about generalizing from, of course, from a single experience, but that's an interesting idea. Now, of course, from these donors, we had isolated many antibodies that were IgG1, and so we can identify the clonal families that produce these antibodies. And these clonal families are generally small, and you see here that these antibodies are very mutated, and a single or just a few clones are maintained in the memory pool. So there has been a lot of selection in these antibodies that are directed against RSV, tetanus, or measles virus, and are mainly IgG1, typically T-dependent responses. Also, by comparing these two individuals, we found that, and using a very strict criteria of clonality, which is the sharing of the same VH and VL, we find that there are many families which are shared by two individuals, and interestingly, these families tend to be mainly IgM or IgG2. With different levels of somatic mutation. So it will be interesting to learn more about this specificity of these convergent antibodies. Now, how stable are these families? We can look at the families isolated in 2020 versus those isolated in 2010, and you see that there is essentially a very good correlation in the sense that especially the largest families that are present are found at both time points. So this stability of families in the memory pool is particularly evident, as you may expect, for large families, and we have confirmed this finding using more strict criteria, where we look at the intrasample versus intersample Morosita Horn index, and found that these indexes are essentially comparable, which again suggests a very stable persistence of memory cells. This is certainly not a surprising finding, but is interesting to document the fact that memory cells we can carry memory cells essentially for a lifetime. Now I come to a more to the last point, which is the repertoire of circulating plasmablasts in the steady state. And I want to go back to a paper that we published sometime 20 years ago in 2002, where we were looking at plasmablasts in peripheral blood, and we reported that 6 days after a tetanus boost, you see there is a huge increase in total plasmablasts secreting IgG, and this increase was accounted essentially largely by an increase in specific in plasmablasts producing specific IgG antibodies against the immunizing antigen. However, at any time points during this peak and at later time points, these antibodies against tetanus, toxoplasma, and measles were always found in circulating plasmablasts at very low frequencies, but in a steady, also in the steady state. So antigen-specific peak on day 6 after boosting, but plasmablasts of different specificities are continuously produced at low rate. And we modeled this and suggested that this could be a mechanism to maintain serological memory. Now, with better technology, we can revisit this data. And so we sort the circulating plasmablast, and these are examples from the 10x data. So first of all, plasmablasts can be easily identified by the fact that they have a 100-fold higher level of antibody transcripts as compared to memory B cells. And this is the UMAP of the circulating plasmablast. These two clusters represent Ki67 positive, so very recently generated plasmablasts. But the point here is that if you look at the distribution of clonal families within those plasmablasts, the distribution is more or less random. So this suggests that, in fact, the heterogeneity that you see here is a micro-heterogeneity of unknown significance, but essentially this is a very homogeneous population of recently generated plasmablasts. So this is, we think, is interesting because it reflects the ongoing activity of the immune system. So we are asking, what does the immune system do when it's doing nothing, essentially when in the steady state? Okay, so these are the representation of plasmablasts in the steady state. So you see here in the center, there are some very large bursts of plasmablasts, which are clonal. Again, mainly IgM and IgA, as you see here on the right. These are all plasmablasts. There are mixed isotypes, and even with exactly the same antibody sequence, as you see here, since they are aligned vertically. So large bursts, mainly IgM, IgA, and IgG2, again, look like T-independent, many singlets, multi-isotype bursts. Now, what's the relationship of these plasmablasts with long-term memory cells? And this is my last really experimental piece of data. Now, if you now look at the plasmablast families found in 2020, you find that 17% of them correspond to memory B cell families found in 2010. So this means that long-term memory, that 17% in this individual and 20% in this individual of long-term memory cells are firing at any given time point. So we thought that this was a particularly interesting finding, and even more interesting is the finding that in these two donors, whenever you look at plasmablasts, you find recurrent plasmablasts that show up in this case, every time that you bleed this donor, there are these 10 plasmablasts or these 4 plasmablast families that produce antibodies. And in this particular donor, these 10 families, which represent plasma cells isolated at different times, as you see here, are primarily IgG2 or IgA or IgM. Once again, T-independent isotopes. And so what about the small IgG1 families? Then we had to look specifically for them by plating a large number of plasmablasts, screening the supernatants, finding specificities like tetanus, measles, and isolating monoclonal antibodies, and formally showing that in the absence of any antigenic stimulation, there is a continuous production of IgG1 plasmablast against recall antigens. And so, just to summarize this last piece of this experiment, large multi-isotope and convergent IgM, IgA, and IgG clonal families, stable memory pool, and these recurrent plasmablasts, which are derived from memory B cells that suggest that either there is persisting antigen stimulating these responses, or that memory cells in some way responded to some generic stimuli and spill out continuously plasmablasts that maintain serum antibody production and replace those long-lived plasma cells which are present in the bone marrow. And with this, I want to show this slide, which here is all the extended laboratory. Now, this is our small laboratory in Milano at the IMGM in Milano. Still, we enjoy a very fruitful collaboration with the laboratory of Federica Sallusto in Bellinzona and Zurich. And I have to acknowledge in particular for the work on the B-cell repertoire analysis, the work of Ganesh Farg. And this is a retreat. You see happy people smiling in the sun. Here we are on the Ligurian coast, and we have brought people from Zurich, Bellinzona, and Milano. These are happy students and postdocs. And once again, I think the best suggestion for young people is really to have fun. So thank you.
Thank you very much, Antonio, for a fantastic journey through B-cell biology and antibody responses. So let me remind everyone that next week, global immunospeaker is going to be Michael Zixt, and that you can, as always, ask questions for Antonio Lanzavecchia via Twitter. You should search for the account Global IMMUNOTALKS. You should find a tweet that says, ask questions for Dr. Antonio Lanzavecchia here, and you can reply to that tweet with your question, and please mention the hashtag Global Immuno in the comment. And Antonio will reply directly from the ImmunoSpeaker Twitter handle. So Antonio, thanks again for this great talk, and see you soon. Bye.